Hello and welcome to the Onion IQ podcast. I'm Alice Formiga of eOrganic, and today I'm talking with Laurel Dunn and Kumar about food safety in organic onion production. This podcast is brought to you by the USDA NIFA OREI project, Onion IQ, improving quality, resilience, soil health, and food safety in organic onion in the Southeastern and Southwestern United States. The project is led by Dr. Bhabesh Dutta, also at the University of Georgia. So today, I'd like to introduce Laurel Dunn and Kumar, who are both associate professors in the Department of Food Science at the University of Georgia. They're going to introduce themselves and their roles in the project and give a short presentation, and then I'm going to ask them some additional questions about how to manage food safety in organic onion production. Okay, so Laurel, take it away.
All right, thank you for that introduction. So as introduced, I'm Laurel Dunn. I'm an associate professor and actually an extension specialist as well, which has been a lot of my involvement in this work. So I got brought in to do some of our applied field studies pertaining to food safety on this project. And then I'm also helping with some of the extension outreach component of this work that we've been doing now for the past couple of years. And so where we got involved with some of this food safety work is some of our previous projects have actually looked at survival of some foodborne pathogens, or at least some surrogates for foodborne pathogens in onion production soils. Because around 2020, we started having, for kind of the first time, some foodborne outbreaks that got traced back to onions. Before that time, we weren't really seeing much illness associated with them as a commodity. And then we had several subsequent outbreaks, particularly with salmonellosis and contamination on onions. And so we started out, and again, this was a previous study where we were actually using E. coli, and we're using it as a surrogate for salmonella because we can't be spraying salmonella out in our production fields, but we can this E. coli strain that doesn't cause illness. And so we were applying it in organic production soils. We were managing those soils the same way they would be if they had onions growing in them. And we found that E. coli could actually survive well beyond the National Organic Program recommendation or requirement for that 90 to 120 day application to harvest interval. And if you're not familiar with those NOP guidelines, what that means is if you're using a raw manure, like a raw chicken litter, which we use quite a bit in Georgia, you're applying it to your production fields. E. coli at least is able to survive beyond that 120 days. And so that 120 days is the time between when that manure is applied and when you actually harvest that crop. And so the 90 days comes in if you're harvesting a crop that doesn't touch the soil. For onion, it obviously is in close contact with the soil. So we have 120 days between application of a raw chicken litter and the harvest of that onion. So that was kind of what got us down into this study, down the path we started to explore, because, all right, we know we can get this, the surrogate survival well on to about 140 days even in some production soils. Are there some mitigations that we can introduce to these soils in order to reduce some of this foodborne risk in our onion production? So what we started looking at was mustard green biofumigation, which is an organic production process where you're growing mustard in fields that you're going to be growing other commodities. You're tilling in that mustard, and some of the chemicals in those plants are actually able to control. We've seen it with some plant disease. We've seen it with other pests that might be a problem in onion. We're interested in, does that same principle work for controlling foodborne pathogens? And so we first started with a lab study where we took some of our organic onion soils or soils that we're using for organic production. We put them in microcosms. In this case, they were little pots that we put in a chamber in the lab and kept under controlled humidity and temperature requirements. And we inoculated these with salmonella. We then took mustard that was grown from these production soils. We mimicked what a tilling process would be and incorporated it into the soil to see the impact of the salmonella due to this kind of mock biofumigation that we were doing in the lab. And what we really found was, this graph kind of shows, is that all of our mustard cultivars really performed about the same as our control, or even a little worse than our control in achieving reduction of salmonella in the soil. So not exactly what we were going for. We were a little discouraged, but our thought was, well, maybe our scale's too small. Like maybe if we do this in a field environment where we're getting more mustard incorporated, we're getting maybe deeper incorporation in the soil, we might see some different results. And so what we ended up doing was taking this to some of our organic production fields where we were planting this mustard. We then used our favorite little E. coli strain again, because again, we can't be putting salmonella out in the environment, particularly on a production farm where people are going to be eating stuff out of that farm. And so we sprayed that E. coli in the field. When the mustard was flowering, which was when we're expecting the highest levels of those compounds to be present. And then we immediately went back over, we flail mowed, we tilled that material into the soil. And then some of our plots we covered with plastic mulch to try to trap in some of those compounds. And then some we just left open to see what sort of control we got from those treatments. So unfortunately, we kind of saw a similar impact as we saw with the salmonella study in the field. Whereas you can see this graph to get you kind of associated with this. We replicated this across 2 years. Our first column is where we just had the mustard. Our second column is where we had our mustard and plastic. And then we have our control that didn't have any mustard or anything tilled in. And so as we can see, these kind of on the left, far left of each of these is our starting inoculum. So we start about 6 log CFU per gram in the soil. And we really found that, first off, we got an interesting spike of some of our populations, but we really didn't see significant drops as we expected across about 21 days in these production environments. And again, as you can kind of see, our control plots almost had similar reductions. So we weren't really seeing a lot of efficacy from this as a management practice. It's not something that we would go out and really start recommending to people from a food safety perspective. And so there might be other parts of the study that have shown some other things, but coming from the food safety side, I don't know that we could really recommend doing this to reduce some of that risk. So we completed a study this fall, and we're actually still going through some of this data, and I can see the project kind of shifting in this direction, and maybe some subsequent projects going in this direction. All right, we don't necessarily have a way to control the foodborne pathogen once it's applied to the soil. So really, our biggest tool is not applying it to the soil in the first place. But if we're in a situation where maybe we're relying on organic fertilizer sources. So we're in Georgia, poultry litter isn't going away. It is a really good way to use that nutrient source. It's great for our poultry industry to get rid of it. And it's great for our farms to get a good nitrogen source in the soil. So are there other ways that we can maybe reduce some of our risk? And we're looking into this by seeing, like, does varying some of our nitrogen application rates and really timing, does that have an impact on some of our pathogen survival? So are we doing maybe late nitrogen applications, or maybe not just focus on nitrogen, but focus on other macro or micronutrients that might be either spiking some of our foodborne pathogen populations, or are there things that we can be adding that might be reducing some of them? So some of our other studies have shown that actually some of our synthetic fertilizers, when applied throughout the season, reduce some of our foodborne pathogens. And we haven't really figured out mechanistically what's happening in those situations, but we're interested in kind of seeing what is happening with this prolonged survival, and then why are some of these synthetics maybe reducing some of that survival. So that's kind of the state of our field studies at this point. And then I can maybe stop sharing here and hand it over.
Okay, thank you. Govin, you can go ahead and share your screen.
Okay, hi. So hi, my name is Govindaraj Dev Kumar. I'm an associate professor at the Department of Food Science and Technology as well, but I am at the Griffin campus. So I work in the Center for Food Safety. And so my role in this project is to actually throw salmonella around in the lab, and we are working with the outbreak strain that caused one of the onion outbreaks. and actually find out, or at least have experiments to find out what's the mechanistic activity that results in salmonella persistence in soil and its cross-transfer to onions. So some of the experiments that we have done is we sequenced the onion outbreak strain of salmonella, and we looked for the alt cluster. And I'll explain what the alt cluster is in the next slide. We looked at some soil survival studies similar to what Laurel had done on the field, but we did it with salmonella and compared it to another strain of salmonella that we had isolated from manures, from cattle manure. So we call that the bovine isolate. And then we looked at soil persistence studies where we tried to understand how the addition of poultry litter could result in biofilms that salmonella could form. And then finally, we looked at mechanisms. Right now, we are looking at mechanisms of soil to bulk transfer of salmonella. So some study overview for our soil survival. So we'll start with the assessment of all genes. So all genes are genes, these are thiosulfonate reductase genes that certain microbes such as Pantoia ananthus has. And so this is a bunch of, it's a cluster of genes. It's around 11 genes that these organisms have to break down thiosulfonates that onions produce, and those are the compounds that make you tear up when you slice an onion, and they have antimicrobial properties. So we wanted to see if our outbreak strain had putative genes that were similar to this. So we sequenced our strain and then we looked for these genes, but we did not find that in our salmonella in the outbreak strain. So and then what we wanted to do was we wanted to see other mechanisms by which we could understand how the bacteria might have got onto onions. So the first thing we did was we looked at soil survival. So what we did was we put in a gene that makes the bacteria actually fluoresce, and then we put it in soil and we grew it we let it survive in soil for 30 days and enumerated it at different time points. We again compared this to a bovine isolate. And what we noticed from the study was that the onion outbreak strain really did not survive in soil as well as the bovine strain. And this could be one because it has these restriction enzymes that chop up the plasmid. And so we might be losing fluorescence because of that. And that's kind of common in wild type strains. So we don't know the mechanism of action, but we know that it really doesn't survive in soil as well as some other isolates of salmonella. So that's another interesting insight that we got. So then we wanted to understand then, well, what could make the bacteria persist in soil? So as Laurel had mentioned, chicken litter is a very popularly used soil amendment, not only in Georgia, but across the United States. So we looked at different types of poultry-based soil amendments. These included poultry pellets, organic poultry pellets, and then just used poultry litter, and then fresh poultry bedding. And we compared all of these for their ability to induce biofilm formation in salmonella. And what was interesting from the study was we found that our salmonella Newport isolate that had caused the onion outbreak actually formed a lot of biofilm, actually formed the highest amount of biofilm compared to any of the serotypes that we looked at in presence of used poultry litter. It also produced pretty high amounts of biofilm in presence of poultry pellets and organic poultry pellets. So that might be one mechanism through which the organism might establish persistence in soil. And then now we're looking at cross-transfer mechanisms. So yeah, it can be present in the soil, but how does it get onto the onion or maybe even inside the onion? So one way we are looking at is if the soil temperature on a summer day is really high and your irrigation water is cooler, does that cause the irrigation water to get sucked in and pull in the bacteria? So that's an ongoing study that we're doing right now. So some of the results that we found, and I touched upon this in the previous slide, so if you look at the first left of the top left of the slide, you can see that those are the fluorescent strains that we developed. And then we mixed them with soil and we looked at survival over a 5-day duration. And over a 5-day duration, what we found was our Salmonella Newport, the onion isolate, seemed to survive have the least ability to survive in soil in comparison to all the other isolates. And when we did a 30-day study, we found that after day 10, the population of Salmonella Newport starts diminishing quite a bit. And then if you look to the right, the top right, that's the amount of biofilm that was formed. So biofilm is this sticky matrix that Salmonella and other types of foodborne pathogens produce, and this helps them aggregate and survive in harsh environments. And what we noticed was, if you look at it says Salmonella Newport onion, the UPL, which is the used poultry litter, induced the highest amount of biofilm. This was followed by poultry pellets and then organic poultry pellets. So all these induce quite a high amount of biofilm production by the pathogen. In comparison, we have Salmonella montevideo. This has been associated with cantaloupes, and we see that in Salmonella montevideo also poultry pellets and organic poultry pellets seem to induce biofilm formation. So that gives us a little bit of an idea of maybe a mechanism through which the pathogen can induce persistence, and it kind of it works in tandem with Dr. Dunn's hypothesis, where she looked at E. coli survival in soil as well. And finally, this is to the bottom right. This is some of the experimental work that we're doing, where we are using dyes and to look at internalization. So what we did was we took an onion, we put it in an incubator, warmed it up to like 40 degrees centigrade. So that's like a really hot summer day, and then exposed it to cooler water, irrigation water that had a dye in it. And when we did that, we found that the dye seemed to find its way inside the whorls of the onion. And so we are now doing experiments with actual pathogen to see if it can also get into the onion just like the dye did. So these are some of the experiments that we're doing. The summary of our findings are that the Salmonella Newport onion outbreak isolate does not have an alt cluster. So it's not surviving inside onions because it's tolerant to the antimicrobials that onions produce. It does have lower stability of the GFP plasmid, and this is because it's a wild type strain. It's out in nature, and it has these enzymes to break down foreign DNA. It has less persistent survival in soil in comparison to the beef isolate. But when we add poultry litter to the soil, it really improves its survival because it increases its ability to form biofilms in presence of poultry litter. And finally, right now we are looking at soil and irrigation water temperature differentials as mechanisms contributing to the internalization of the pathogen inside onions. So yeah, that's an overview of some of the projects that we're doing right now.
So I guess I just had a question about what you just were talking about. Like you were saying that its survival isn't as long in the soil, but it can still cause major food safety outbreaks, right?
Yes.
Yeah.
What we are noticing, at least in the lab, is compared to another strain that we had isolated from cattle manure, it seems to not survive as well. But that also might give it the impetus so there might be some mechanistic action where it persists. And that's where we realize that when we add poultry litter to the soil, this forms a lot of biofilm, and that might help it persist in soil better. So it might, on its own, it might not do too well, but when you add poultry litter, there seems to be it seems to help it persist better.
Okay, so what would you recommend that growers do instead?
That's a good question, but initially we first want to find out. This is just one study where we have seen, and it's an in-lab study where we have seen that poultry litter can induce biofilm formation. But we might want to do more studies to see, do these biofilms help it attach to soil particles better and persist in soil better? When we do more studies, we'll find out. But obviously, some of the solutions is composting. Composting can help. We looked at two parameters. We looked at bile as a biomarker in poultry litter, and we found that when there's a high amount of bile, it kind of correlates to the amount of biofilm that the pathogen produces. And then some of these poultry pellets also have a little higher amount of sugar in them. So, and that's because sometimes they use binding agents that might have sugar in them, like molasses or something, and that might also contribute to better survival. But all I have is hypotheses. These need to be tested out.
Okay, yeah, that's interesting. So, how can growers assess their safety risks in the field?
So some of the things, and Govin and I think both want to be really clear, we're not advocating for stopping the usage of poultry litter or some of these poultry-based products. What we're trying to do is increase some awareness that you can have some enhanced survival occurring, and really trying to discourage practices like side dressing. So side dressing with untreated or uncomposted types of materials that might be introducing some of these increased populations of these pathogens. So really making sure that we're adhering to that 120-day application interval is going to be a huge way to make sure, yes, we saw some of that extended survival to 140 days, but we were putting really high levels of these microorganisms in the field in the first place. So from a public health perspective, most instances would not be introducing those really high levels. As Govin said, composting using some of these treated materials might be a way to reduce risk, although we're seeing kind of some funny data with some of these heat treated poultry pellets where we're seeing some spiking of some of these populations. And really just good practices in excluding wildlife, and managing the packing environment and where things are being cured to make sure there's good cleaning and sanitation in those environments are really the best things that anyone can be doing to reduce some of this risk.
Yeah, maybe you could talk a little more about that. That was definitely one of the questions of, what kind of proper storage conditions can help maintain quality and food safety.
Govin, do you want to jump in, or
Laurel, why don't you start? Because yes, yeah, why don't you start, and then I'll try to think of something smart to say.
So I mean, storage, it's one of those things, you are pulling this from the soil. So you have a good amount of soil going into your production environments. And we do see this as a little bit of a problem, particularly for firms that might be doing some sort of environmental monitoring, where they're looking for foodborne pathogens in their packing house or in their storage facility. They're looking a lot of times for pathogens that are found in the soil, and they're pulling a crop from the soil, and a lot of times introducing it to their equipment. And so making sure that, keeping up with sanitation protocols regularly on their equipment before they're doing any environmental monitoring, because that monitoring is really looking for some of those persistent populations in their storage areas or on their packing equipment in the first place. So making sure, like, if environmental monitoring is going to be a part of their food safety program, which we're seeing it happening more and more in our packing houses in general, making sure that what you're doing before you're implementing those sorts of programs, so you don't get yourself in trouble by mixing up when you sanitize and when you collect a sample, for instance. But really just keeping things, keeping a clean house, keeping things cleaned and sanitized, keeping dirt out of storage areas, trying to limit as much as possible how much bins or containers or things like that are placed directly on the ground. Once we pull it out of the ground, once we start knocking the dirt off, we want to make sure that we don't have additional opportunities for recontamination. With produce, we tend to forget, because it does grow outside, that it's still food. While it's growing on the plant, it's still food. When we harvest it, while it's curing, it's still food. Unless you cook it, if there are foodborne pathogens on it, you're not getting rid of them. And so you need to treat it every step of the way, like someone's going to put it in their mouth without doing anything to it, because that's what a lot of us do with fresh onions. That's what I just did for lunch. So really treating it like food the entire step of the way.
Okay, are there any emerging technologies that help detect or prevent contamination?
Laurel, do you want me to go?
Go for it.
Sure. So, I mean, also adding to what Laurel said, right? Once the onions are harvested, there have been studies that have shown that foodborne pathogens can survive on, chopped onions. So when they reach retail and you buy like chopped onions, it's very important to see that they're using the sanitation that is required, and then it's maintained at the right temperature. And also consumer awareness. Like Laurel said, you're eating a product that grows in the soil. And so, not if you're finding something that's spoiled or slimy, or has even the black film of a mold growing on it, like don't consume it, because these are high in nutrition and even sugars, and they could host not only spoilage bacteria but also foodborne pathogens. And then some of the intervention methods during curing. So curing is when you pull the onions out of the soil and then you let it dry. So that's an important step where we could intervene. So seeing that there's less moisture, there's no rainfall, like choosing the right day to harvest your onions, and then storing it under the right conditions. And then there are new intervention technologies like antimicrobial lights that people are exploring, UV lights, but it comes down to the basics, preventing it, its contact with soil surfaces, basically.
Thank you. Just one more question here because we're running low on time, but I just was wondering if you could talk about your future studies and what you plan to do next based on the outcomes of these first year trials, or year 1 and 2 trials.
Laurel's made a lot of progress with the field studies. That's kind of given me a direction to explore and see how I can take this further. So we're looking to see if these poultry amendments can result in biofilm formation on soil particles, and that'll give us an idea of what is causing persistence mechanisms and what biomarkers can be used to improve, to actually assess the safety of these amendments. One interesting thing that we found out when we were doing our study was what we would do is we take these poultry amendments and make extracts out of it, and we found a contaminant growing in our extract, and that contaminant actually uses bile, reduces the amount of bile in the amendment. So we don't know if that could be used as a biocontrol agent while we're doing composting, but just I guess those are interesting areas that we're exploring at the moment.
Okay, yeah, that does sound interesting. Laurel, do you want to let us know what you're up to in the next couple of years?
Yeah, and I had touched on it a little bit at the end. We're probably going to continue on the route of some of those application intervals, and really trying to elucidate what is happening, both with our organic amendments, those manure-based compounds, and some of those synthetics, since we are getting, suppression of pathogens with some of those, and we're getting increased, or at least increased persistence and survival with some of those more organic amendments. So really trying to dig into what is occurring there mechanistically.
Wonderful. Okay, this sounds really useful, and I'll look forward to hearing more as it progresses. So thank you very much, both of you. And the recording of this will be available on the eOrganic YouTube channel. So you'll be able to find that there, along with some of the other ones that we've done reviewing the different aspects of this project. So thank you both very much.
Thank you.
Thank you.
Thank you.